and thank you so much for the invitation to speak today. Hopefully you like strange venomous animals, because I'm going to tell you about what I think is one of the coolest and weirdest, the platypus. To begin with, I'd just like to acknowledge that tonight I am speaking from the land of the Gadigal people of the Eora Nation, and I'd like to acknowledge their elders and pay respects to them, elders past and present. And this is a photograph of some rock art of a platypus in Cape York in Northern Australia. I would also like to acknowledge that I've been very fortunate to work with a range of fantastic collaborators on the research that I'll present to you today, in particular Kathy Belov at the University of Sydney and also Wes Warren at the McDonald Genome Institute. I'd also like to thank my lab, although the Whittington lab works more on pregnancy and the placenta at the moment, and also our funding sources. So in my lab, what we're really interested in understanding is evolutionary innovations. So these are traits like these, dramatic, really adaptive traits. They've led to significant consequences for the life histories of these organisms. And often they're very difficult to study, and it's difficult to understand how they got there, because in many cases, there are no transitional forms available to study. And also these complex traits are controlled by interconnecting networks of thousands of genes that co-evolved together. And it's only recently that we've been able to use genomic tools to start to untangle them. And of course, they've been really useful in untangling the evolution of complex traits like venom and also pregnancy and the placenta, which is what I work on most at the moment. So the evolutionary innovations that we use tend to be innovations that have evolved convergently, so multiple times in independent lineages. And the reason for that is they give us really wonderful examples of naturally replicated evolutionary experiments that we can use to test our ideas. So one of the questions that we are asking is, are convergently evolving phenotypes like venom underpinned by similar genes and similar evolutionary mechanisms? So I'm sure that I don't need to tell this audience that venom is a really fantastic example of a convergently evolving trait. It's evolved many, many times in invertebrates and vertebrates. We do have just a few venomous mammals. And one of those, of course, is the wonderful duck-billed platypus that I'll tell you about today. So a crash course on monotremes. It's not very long. We don't have very many monotremes on the planet today. We have the Australian platypus. We have the short-beaked echidna, which is found in Australia and Papua New Guinea, and 3 species of long-beaked echidnas, which are found in Papua New Guinea. Now, we know, though, that from the fossil record that monotremes used to be a lot more diverse, and these are just some representations of some of those fossils. The one in the bottom right-hand corner is probably my favourite. That's the Glossus hacketi. It was an echidna-like animal, sheep size, which must have been very impressive. But really the take-home message from this slide is that ancient monotremes were a lot more diverse and a lot more widespread than the monotremes that are on the planet today. And we think that echidnas had originally amphibious ancestry. So it's probable that the common ancestor of echidnas and the platypus was a little bit more like a platypus, and we'll come back to that later. So because these animals are monotremes, egg-laying mammals, they're the earliest offshoot of the mammalian lineage. They have a number of characters that are pretty plesiomorphic. So things like quite a reptilian skeleton. This is a platypus skeleton, and you can see the legs are quite splayed out, and that's going to be important later. And the reason for the splaying of the legs is that they have quite a reptilian shoulder girdle and pelvic girdle. They're also egg-laying, of course, but they have many, many features that put it very firmly in the class of mammals, which is they have fur, they produce milk to nourish their young, although they don't have teats, and they also regulate their body temperature. So the timescale that we're talking about is the monotremes diverging from the rest of the mammals about 186 million years ago. Now, clearly the platypus is, a unique Australian animal, but it's also very under-researched. And I think that's really mostly down to the fact that samples are very, very difficult to obtain. It's also extremely difficult to breed these animals in captivity. Until about 5 or 10 years ago, there had only been 4 or 5 births in captivity of platypuses. And they're also hard to study in the wild. They're very cryptic. They're active at really odd times of the night. I work on reptiles now, which is much more fun to catch. So it's just really difficult to study platypuses. But genomics came along, and that was really important because it suddenly gave us this opportunity to understand more about the fundamental biology of these unique animals. So we sequenced the genome of the platypus, and this was quite a long time ago in 2008. It was one of the first mammalian genome projects after the Human Genome Project. My role in that was to look at immune genes and venom genes, which are actually related, as I'll tell you about today. So a little bit about the weird and wonderful venom system of the platypus. This is the hindquarters of a platypus. So here we have a tail, the hind leg, and the venom gland is in the thigh area. It's connected via a duct to this thing here, which is called an extratarsal spur. Now, the venom gland is also called a crural gland, and it is thought to have had an origin from a sweat gland originally. And the extratarsal spur is a really interesting structure. So as you can see in this sort of dissected specimen, it's very sharp, it's hollow, it's keratinous, and it's also sitting on top of a bone called the os calcaris. Now, this os calcaris is pretty important because what happens is that this spur is actually able to articulate against that underlying bone, and it allows platypuses to actually erect their spurs, which when they're not erect, they just flop around, erect their spurs so that they're rigid and they can actually inject venom into a victim. So remember this little bone because we'll be coming back to that later in the talk. Now, the very strange thing about platypus venom is that it is sexually dimorphic. So we only have the venom delivery system of this mature spur and venom in adult male platypuses. Females hatch out of eggs and they have these tiny little vestigial spurs, but they drop off. So we've got venom only in the males. And the other really unusual thing about the venom is that it's only produced in the breeding season, which is quite strange. And it leads us to speculate that the function of this venom has probably got something to do with reproduction now, because platypuses don't have any native predators anymore. Now, we have a hypothesis about what the venom functions for in the platypus, but this still hasn't been conclusively proven. So it just indicates how little we know about the biology of this animal. So what we think happens in platypuses is that males use their spurs and their venom to defend their territories against other male platypuses during the breeding season. Now, there's several lines of evidence for that beyond the fact that only males have venom and they only produce it in the breeding season. That's the fact that we have trapped male platypuses with healed spur marks quite often. And the second line of evidence is that one male platypus has been trapped with a very new, what looked like a spur wound, and it was actually paralyzed in that particular area of its body, and the paralysis was temporary, so it wore off later. So you could imagine that there would be quite a big adaptive advantage if you're a male platypus to temporarily disabling your competitors and monopolizing access to females. So that's our best guess about what platypus venom is for. Now, of course, platypus venom could also be deployed very successfully as a defensive weapon. And that can be against domestic dogs or foxes, and also very rarely the occasional unwary human like a fisherman or a researcher. Fortunately, not me. Now, I say fortunately because by all accounts, platypus envenomation is very unpleasant. You'll get swelling of the affected area and also a really extreme pain. It's systemic. It's very long lasting. So we're talking about 2 to 3 weeks of extreme pain, and normal painkillers have no effect against that pain of platypus envenomation. Even morphine doesn't have an effect. The only treatment is a nerve blockade. Now, there's one case report of platypus envenomation in a Vietnam War veteran who actually, I believe, got a medal for bravery, and he sustained terrible shrapnel injuries in the war. And he described the pain of platypus envenomation as far, far worse. So clearly, these are pretty strange symptoms compared to envenomation from other animals. And it makes us think that there may be some pretty interesting, unusual toxins in the venom to be causing those strange symptoms. Now, while it was thought before we started this work that there were maybe 50 toxins in the venom, only 3 of them had ever been identified. So this is part of the fundamental biology of the platypus that was almost a complete mystery. And that's really what motivated my work. So what we were interested in understanding was, firstly, what is in platypus venom? Might it contain clinically useful substances? And can we figure out more about the fundamental biology of this animal? And my passion really was to understand the mechanisms of evolution in venom in the platypus. So we decided to identify venom toxins in this animal. And I should say that I'm just going to use the generic word toxins to refer to the peptide components of platypus venom, which are the most abundant components. Unfortunately, we don't really have the luxury of large quantities of venom to study from platypuses, and that's because only males produce the venom. They produce very small quantities, so maybe 70 microliters at a time. And they also, because they don't breed well in captivity, keepers are understandably reluctant to disturb animals during the breeding season to actually collect the venom. So consequently, that wasn't really an option for us. And instead, we had to take a step back and look at the genome and also the transcriptome of the venom gland. Now, even the transcriptome was quite difficult. We spent quite a bit of time waiting around for an opportunistically collected sample. And then very fortunately for us, but unfortunately for the platypuses, we got an animal that had been hit by a car and an animal that had been killed by a dog. They were both retrieved very quickly, and they made it to us in time to actually get high quality RNA. And that allowed us to do a transcriptome project. So we took advantage, there were 2 papers. In one of the papers, we took advantage of the differential production of venom across the year in the platypus. So on the x-axis of this graph, we have the month of the year, and the y-axis is the venom gland weight per kilogram of body weight. And what you'll notice is that the venom gland regresses in the winter, Australian winter, and then it hypertrophies in Australian spring. And when it hypertrophies, it's secretory, it's producing a lot of venom. So we're able to have a sample from October and a sample from March, and actually look for up and down regulation of genes between those 2 time points. And what we did was we were able to identify 88 new toxins from 18 different families with homology to venom toxins in other species. And also taking advantage of this differential production of venom, we were able to identify 5 completely novel toxins, which potentially might be involved in producing the pain of platypus envenomation, inflammation, maybe neurotoxic effects, and possibly also synergistic effects with other venom components. So that's about all I'm going to say about what's in platypus venom. I now want to take you through the story of venom gene evolution in this animal. So how did the toxins actually evolve in this species? And to do that, I'm going to focus on one family of toxins, and that's the defensin-like peptide family, or the DLPs. The reason I chose to concentrate on these peptides was that it was confirmed at the proteomic level that these were the most abundant peptides in the venom. But they had an unknown role. The only thing that we did know about them was that their amino acid sequence was quite similar to the amino acid sequence of some peptides called beta defensins, which you may have come across in your own work. These are peptides of the innate immune system, and they're antimicrobial, so they'll actually interact with bacterial cell walls to kill the bacteria. And if you look at the folding structure of a platypus venom defensin-like peptide in grey and a beta defensin in black, you can see that they have a very similar folded structure. And that led us to believe that potentially these 2 classes of peptides might be evolutionarily related. So the first thing that we decided to do was to mine the platypus genome for the sequences of both the antimicrobial beta defensin genes and the venom defensin-like peptide genes. And this is just showing a partial amino acid sequence alignment, showing you the most important bit of those peptides, which is the part of peptides that contains 6 conserved cysteine residues. You're probably aware that cysteines are very critical in producing the folding structure of a protein, because they actually form disulfide bonds and help a protein fold up into its proper shape. So the 6 cysteines are present in both the antimicrobial peptides and the venom peptides, but we did notice that there is a slight difference in the spacing of those cysteines. So there is a 4 amino acid insertion and a 2 amino acid deletion in the venom peptides, which suggested that maybe they had slightly different folding structures. Now, if you have sharp eyes, you will have noticed that there is a peptide in the middle here that didn't get classified as either a beta defensin that's antimicrobial or a venom defensin-like peptide. And the reason for that was that it had characteristics of both. It didn't really fit into either category. And we started calling this the intermediate peptide for reasons that hopefully will become apparent as I take you through this talk. Now, at the time, we didn't have the beautiful long-read genome sequencing that we have access to now. So we actually didn't know where these genes were sitting on the platypus genome from our genome assembly. So we did fluorescence in situ hybridization to actually locate each one of these genes onto the platypus chromosome. And what we found was that these defensin-like peptides, the antimicrobial ones, they were found in clusters all over the genome, which is exactly what we'd expect based on the genome sequences of other animals. But one of those clusters was particularly intriguing because it contained a beta defensin, all 3 of the venom defensin-like peptide genes, and the intermediate gene. And in fact, these genes were all extremely close together on the chromosome. So only about 50 kilobases separating them all. And their arrangement is really a hallmark of a process called gene duplication, which of course is where we have a single copy of a gene. At some point in evolutionary history, it has been duplicated accidentally. Normally, that one of the duplicates would just accumulate mutations and become non-functional, but very, very occasionally, it can accumulate mutations and take on a new function. And that's what we think has happened with these defensin-like peptides. We've had multiple rounds of gene duplication from an original beta defensin gene. Now, that's also not the whole story of platypus venom gene evolution. So it's important to note that there are other venom genes in the platypus that haven't evolved by gene duplication. So alternative splicing, mutation, and also co-option of single copy genes are likely also to play a role in the components of platypus venom. We were able to date the duplications of the defensin-like peptide genes in the platypus in work that was led by Emily Wong. And we found that the intermediate gene was duplicated about 192 million years ago. And the first defensin-like peptide copy appeared about 98 million years ago. And I will come back to this dating of these gene duplications a little bit later on as well. So we have this idea that there's been some co-option from non-toxins to form this defensin-like peptide family. We've had a beta defensin, it's been duplicated, we've ended up with multiple venom genes. Now, we know that the beta defensin is antimicrobial. We know the defensin-like peptides are toxins. We're not really sure about the intermediate peptide. So we decided to actually functionally test these peptides by synthesizing them artificially and then just testing them on a standard bacterial killing assay. And what we found against E. coli was that the beta defensin was strongly antimicrobial, exactly as we would expect. The defensin-like peptides from the venom were not antimicrobial at all. And this intermediate molecule had some very weak antimicrobial activity. Now, the reason that we think that the beta defensins are antimicrobial and the venom defensin-like peptides are not is down to the spacing of the cysteine residues that I showed you in their amino acid sequences. So when the beta defensins fold up, as you can see in this model, we have a separation of the hydrophilic and hydrophobic blue and yellow residues, whereas in the venom defensin-like peptides, in part because of that slight difference in the cysteine spacing, we don't end up with that clear separation of blue and yellow residues. And we think that that means that these venom defensin-like peptides just can't interact with bacterial cell walls to actually kill the bacteria. Now, we also did an assay of gene expression across a panel of tissues of these genes. So this is kind of old school, but if you haven't seen one of these, each column represents a different tissue. Each row represents a different gene. So the top row is the positive control. And if there's a band, that means that that gene is expressed in that tissue. And I've also added some dots just because some of the bands are quite faint. So if there's a dot, there's a faint band there. So we did this for all of the beta defensins as well, and we found that they were expressed in different tissues, but not the venom gland. And then when we looked at the defensin-like peptides, we find that indeed all three of them are expressed in the venom gland, two of them very highly expressed, as evidenced by the bright bands. But some of them were expressed in other tissues beyond the venom gland. So you can see this one here is expressed in a range of other tissues, including in this one, even in female tissues. Now, one of them is really solely expressed in the venom gland. And if we look at when those genes actually duplicated, we see a pretty interesting pattern, which is that the oldest defensin-like peptide gene is the one that has the most ubiquitous expression. And the most recent gene duplicate from about 47 million years ago is solely expressed in the venom gland. So we have this idea that potentially there have been iterative rounds of gene duplication that have led to more and more specialized genes that are producing these venom toxins in the platypus. Now you'll notice the intermediate gene down here, it was expressed in every tissue that we tested, including the venom gland. So we're really not quite sure what that gene's doing. We speculate that maybe it's involved in antimicrobial protection in the venom duct, but that's really not been tested. It's a bit of a mystery, that gene. So I'd like to move on a little bit from talking about venom genes to talking about the venom delivery system in monotremes. Now, I've already told you about the platypus venom delivery system, and down the bottom here, you can see what that looks like dissected out of an animal. So nasty spur, venom duct, venom gland. On the right-hand side, we have the venom system of a short-beaked echidna. So you'll note that they do have spur, but spur is actually a lot smaller than a platypus spur. And you really see that in the bottom picture there. You can almost not see that echidna spur. And in fact, when you look at the literature, when you talk to people who work with echidnas, what you find is that no one's ever seen an echidna erect its spurs, and no one's ever seen an echidna use its spurs aggressively or defensively in any way. And in fact, when you look at the anatomy of the echidna spur and that os calcaris, so that bone that sits under the spur in the platypus, you find that in the echidna, they're actually arranged differently, such that we don't actually think that that spur can articulate against the bone. So those spurs don't look like they can actually be erected to jam into the victim. So we have this idea that maybe echidnas are in possession of a regressed venom system. Now, Emily Wong led a study in 2013 to actually assess this using an echidna crural gland. So she sequenced the transcriptome of that gland. And what was found was that there was actually no correlation in the top 50 most highly expressed genes in the platypus venom gland and the echidna crural gland. And in fact, that echidna gland was enriched for all sorts of genes that were involved putatively in steroidal and fatty acid production. So our best guess at the moment for what the echidna crural system is actually doing, based on that genomic data, and also based on the fact that often these echidnas have sort of a waxy exudate coming from the base of their spurs, we speculate that the crural gland is actually being used to produce high molecular weight compounds that might be involved in mate attraction, because we do know that echidnas follow each other around during the breeding season. So it really does look like this echidna venom crural system is no longer having any kind of venom function. And for me, the final nail in the coffin of the idea that short-beaked echidnas might have venom came just a couple of months ago, when there was a very high quality genome of the short-beaked echidna and the platypus that was produced. And I was very pleased to see that they paid particular attention to chromosome X2, which is my favorite platypus chromosome because it has those defensin-like peptide genes on it. And I'll just overlay the schematic that you've already seen from earlier in my talk. So this is part of chromosome X2 that has the platypus 1 beta defensin, 3 defensin-like peptides, and the intermediate gene. And if you look at the syntentic, the same region in the echidna, this is what you find. So we have the same beta defensin. We have that weird intermediate gene that's sitting there still, but we've actually lost 2 of the 3 defensin-like peptide genes. And the only one that's left, it's not even a functional gene. It's a pseudo gene. It's actually lost an exon. So it really does look like that the echidna crural system no longer has any venom function. We have a venomous platypus, we have a non-venomous short-beaked echidna, and that leads us to wonder whether the ancestral monotreme was venomous or not. But unfortunately, a lot of the fossil record consists of things like this. So bits of jaw, teeth, fragments of bone. We don't have an intact ankle that would show us whether the ancestral monotreme actually had a spur or not. However, we can look a little bit more broadly in the fossil record to speculate about whether it was venomous or not. And to do that, we can look at a number of fossils from early mammaliform animals and some non-monotreme mammals. So the leftmost picture and the center picture, they are of two different species of cynodont, which are mammaliform animals, but they're not members of the crown group Mammalia. And quite a number of these fossils have now been found with features in them that have been interpreted as an extratarsal spur. And the same thing has been found in a number of non-monotreme early mammals, like the one on the right. So this is a Mesozoic mammal, not a monotreme. But what you can see is there is a structure on the right there. This is the ankle of the fossil. And this particular structure here has also been interpreted as an extratarsal spur. So potentially venom, and I'm sure there's many people here who've thought more about this and maybe have some expertise in this fossil evidence, but there is this idea that maybe many early mammals were venomous. And if that was the case, then maybe, yes, the ancestral monotreme was venomous as well. So laying all of this information together, the fossil evidence, the gene expression evidence, the genomic evidence, this is our current picture of monotreme venom evolution. And it's a picture that may change as we get additional fossil or additional genome evidence. So what we think at the moment is, if it's true that there was extratarsal spurs and maybe venom to go along with it in ancient mammals, we have in the lineage leading to the extant monotremes, for some reason, the venom becoming sexually dimorphic. So maybe that's because the venom originally had a defensive function and then there were no predators at some point. And so this venom's then been recruited to reproductive function. That's been retained in male platypuses. The spurs have been retained in echidnas, but the venom has been lost. And in the therian mammals, of course, the extratarsal spur has been lost and the venom has been lost as well. Now, if you'll allow me to indulge in some rampant speculation, one could imagine that loss of the spurs might have something to do with the stance of the therian mammals. So I mentioned the shoulder girdle in the platypus. They walk with a very splayed stance. There's actually no danger of them spurring themselves with that kind of stance. But in therian mammals, they have a much more upright parasagittal stance, and you could imagine that it would be quite easy to wound yourself with these extratarsal spurs. So maybe that was why venom was lost. But again, that is rampant speculation. So I'd like to leave you just with the final idea of just touching on convergent evolution again. Quite a number of the platypus venom genes, in fact, most of them, have homologs in venom genes in other venomous species. And that includes the defensin-like peptides. So this schematic just shows that there have been multiple lineages of beta defensins, those antimicrobial genes, that have led, the red lineage has been recruited into venom gland expression in the platypus, in the defensin-like peptides, and the blue lineages have been recruited into venom gland expression in some of the reptiles. So for example, crotamine seems to be evolutionarily related to beta defensins as well. And in fact, it has a very similar folding structure to defensin-like peptides. Now, I think that's pretty interesting because we're seeing that there are convergent venom genes that have evolved, despite the fact that we're talking about venom glands that have different tissue origins. So sweat gland versus salivary gland. They also have different functions. For example, prey capture versus intraspecific competition. So that suggests, as many others have suggested, that there might be preferentially selected peptide motifs that are actually recruited into venom gland expression each time that phenotype evolves. And I'll just leave you with the idea that I think platypus venom really is an untapped resource for biomedical research. As I understand it, there's only been one foray into that in platypus venom, which is where a component's been used as a potential treatment for type 2 diabetes. But the rest seems to be wide open. So I very much hope that someone will follow up on that in the future. And that's about it. I'm happy to take any questions now.